Today we're going to be talking about passive transport. So with passive transport, this hopefully will be a review, and these are all things that you've probably experienced, throughout your everyday life, like when you're cooking or when you're drinking water, things like that. So we're going to be starting with our most simple one, which is going to be diffusion. So for diffusion, what this means is that I have a net movement of molecules or atoms from an area of high concentration to an area of low concentration. So what does that mean? So if I have, let's say, this beaker, okay, and I'm inserting a bunch of molecules, maybe it's sugar molecules, salt molecules, even things like food dye, okay, and I can say like the rest of this is water. Then right here where I inserted these molecules, this is an area of high concentration. They need to spread out so that way they can reach equilibrium. Remember, that's always the desired effect. We always want to reach equilibrium. So a very good example, so whenever you eat food, you have a bunch of food in your digestive system, but that, food doesn't stay there. Those nutrients want to permeate throughout our body. And so some of those molecules are going to be moving through our body through diffusion. And so, it's going to move from an area of high concentration to low concentration. Now, one of the key things for this that we need to remember is that all of these molecules are moving randomly. Okay, so it's all random movement. Okay, so what does that mean? That means that this little molecule, it may be heading this way, but at some point it may head back. Okay, so once it reaches equilibrium, they're all going to be moving back and forth completely at random. So how can we tell what direction it's going to be moving? Well, whenever we're looking at our solution, there is going to be a gradient established. And so what a gradient is when there's a defined area that has a high and low concentration of solutes. Okay, and solutes are just my molecules. Okay, so for example, when I'm looking at this picture that's part of this definition, I could see that I have a high concentration of molecules up here at the top, and then down here not so many. So I could say that I have a gradient right here because there's more molecules up here than there are down here. And so if I were going to have any type of net movement of molecules, they're all going to be moving down. And they will do so until they reach equilibrium. Once they reach equilibrium, I can see that they're all evenly spaced. There's no gradient here. Okay, so all molecules are equally spaced. Okay, but remember, once we're in equilibrium, there's still going to be some random movement. These molecules are going to be moving up and down. So they're just going every which way. Okay, they're just all bouncing around. All right, so that's what our molecules are doing. They're not stagnant. They're constantly moving. And so what can affect this movement? What can affect our rate of diffusion? Well, we have several things. So one, change in temperature. Okay, so if you increase the temperature, these molecules can probably move a little bit faster. Changes in my concentration. Okay, may seem self-explanatory, but the more molecules you're putting into that solvent, then, the faster your rate of diffusion. Also, the solution the molecules are diffusing through. So things like water, okay, so water is very liquidy. It sloshes around whenever you have it in a cup, but if we're trying to diffuse through something like, let's say, oil is extremely viscous, and so it's not going to move around very much. Same thing with, like, let's say syrup. Like, let's say we want to dilute some syrup. If we have a cup of syrup and we pour some water in it and let it diffuse naturally, it's going to take a little while, and that's just because syrup, it's very viscous. That material is very thick, trying to move molecules through. So if we think of some real life diffusion examples, those of you that like bacon, okay, so bacon, that smell of bacon going through the air. I don't know about you guys, but whenever I make bacon at my house, that smell stays around for a long time. So I don't know why. Maybe I need to buy a better air filter. But, that smell sticks around for quite a while. So also, maybe if you, are walking through, like somewhere that's a public place, like when we could easily walk through the mall, or walk around outside, things like that. If you walk past somebody who has a really heavy perfume and maybe they walked by and, they haven't been there, like you don't even see the person, but you still smell that perfume. That's another example of diffusion. So I know, I've been in some offices where it's like, somebody is wearing really strong perfume, and, it would have been minutes after they walked by and you could still smell it. So this is an example of diffusion of molecules. So what does this not consist of? So this, honestly, does not consist of water molecules. Okay, so next we're going to talk about osmosis. So we're going through all the passive transports. Just because you do know passive transport, this is all going to just be a review. And so, this might be a little bit long, but hopefully, it'll jog your memory. So what is osmosis? Well, if we're talking about osmosis, technically, this is the net movement of water molecules across the semi-permeable membrane to an area of higher concentration of solute. Okay, whoa, like that's a big definition. So let's break this down. Okay, so it's a net movement of water molecules across a semi-permeable. Remember, semi-permeable means it doesn't let everything in or out. Membrane towards an area of high solute concentration. Okay, so what that means is that those solutes are going to be things like sugar, salt, ions. It wants to dilute that. Okay, so wants to dilute. So in my picture here, I have a semi-permeable membrane. Okay, so remember, semi-permeable, okay, only certain things go in and out. So in my semi-permeable membrane, my water molecules can fit through. If I look over here, I have these little ion molecules. They can be whatever, whatever you want them to be, but those are too big to travel through my membrane. So the only thing that can move through are my water molecules. I don't have any ions over here. And because the goal for this is still equilibrium, remember, the goal for passive transport is always equilibrium. My water molecules are going to try to move over here and dilute this as much as possible in order to make the sides the same. So what are some things that can impact the rate of osmosis? So, the water molecules moving over here. Well, same thing as diffusion, because osmosis is the diffusion of water. Okay, so changes in temperature. Concentration differences. And, surprisingly, how permeable the membrane is to water. All right, so we're actually going to talk about that on our next diagram. So remember, our end goal is to reach equilibrium. So we want an isotonic solution. So isotonic means that we're in equilibrium. This is where we have a set of solutions that have the same concentration of solute molecules. Okay, and so right here, I could see that I have these two ions, okay? So two on either side, and then I have my water molecules that are clustered all around. So it's very important to remember that water, because it can fit through the membrane, water is going to be moving back and forth. But in general, when it's in equilibrium, we're going to have about equal amounts of water on either side of that gradient. So you may have heard of reverse osmosis. If you look at the back of a water bottle, most companies say that they purify their water using reverse osmosis. So essentially what that means is companies use a high-power pump to force water through a membrane, okay? And what ends up happening is you purify the water through this, okay? So companies force water through a membrane to purify substances. That's a very strange shaped box to write in. So next, how does this impact us? So if we're looking at our osmosis in our cells, okay, so if you recall, I said small amounts of water are going to go through the membrane. The reason for that is because our membranes are semi-permeable. They don't want to let all of the water in. And so because they don't want to let all of the water in, we're going to have these protein channels called aquaporins. And so aquaporins are where most of our water is going to go through. Okay, so only small amounts of water travel through the membrane. Okay, so for those of you that remember, having a protein channel means that this is no longer regular diffusion, this is facilitated diffusion. You have a protein channel strictly dedicated for those molecules to move through. And so water technically moves using facilitated diffusion, which is going to be our last page of notes. And we're going to be using an aquaporin, which is a protein channel. Okay, so I can see right here through my membrane, okay, I have more water on top of my membrane than I do down here, and I have more solutes. So I'll have a couple of water molecules moving through, but most of them will go through this aquaporin to dilute this side. So I have a concentration gradient here, so I have a higher concentration at the bottom of my cell than I do at the top. So how does this impact our cells? So when we're talking about osmosis effects on cells, we know that water flows in and out constantly. And we know it's completely random. Okay, when I'm talking about this, I'm talking about the molecules. Okay, so the molecules, let me put that right there. Okay, so we still have a net movement. Remember, equilibrium is desired. That's where we want to get. And so if I'm looking at these different animal cells, I have 3 different types of solution. And so when I'm in isotonic solution, everything's in equilibrium. Okay, so I have the same amount of water inside and outside. And so because of that, my water molecules are going to be moving in and out consistently. So I can also have a hypertonic solution. So with a hypertonic solution, essentially what that means is that I have a greater concentration of solutes outside my cell than I do inside. Okay? So because my cell is more diluted, water is going to flow out of the cell. Okay, remember, we want to get to equilibrium, so all the water that's in the cell is going to try to dilute the solution that it's sitting in. And so because of that, my cell shrinks and shrivels. On the opposite side, okay, I could have a hypotonic solution. Hypotonic means that my solution is more dilute than my cell. And so with this case, my net water movement is going to be in to the cell. So the problem with this is that we don't have a cell wall. There's nothing keeping this membrane intact. And so you can see right here, okay, so that membrane opened. And so what ends up happening is the cell bursts, okay, it lyses. And so that's called cytolysis. Okay, so remember I told you lysis means to break, cyto is cell. So the cell breaks, the cell bursts. So these are for animal cells. What happens in a plant cell? Okay, because plant cells have a cell wall. So if I'm looking at a plant cell in isotonic solution, it's the same thing. Okay, so same thing for as my animal cell. Okay, water's going to be moving in and out. Okay, it's at equilibrium. And so water freely moves in and out. Okay, so the problem with this is that plant cells don't necessarily like isotonic solution. If you think about their vacuole, okay, so if they have a huge vacuole, I'm going to use a different color. Okay, so they have a huge vacuole here. You can see that it's probably partially filled up. Okay, ideally, that vacuole should take up most of the space and push up against that plant cell wall. So that way the plant can stay turgid, it can stand up. It's actually going to be part of our lab next week. So when plants get wilty, okay, it's because at this point they don't have water. Technically, they're in a hypertonic solution. Okay, so I can see that there's not much water here. So my cell wall stays the same. Water's going to flow out. Okay. And if I'm thinking about my vacuole is super shrunk. Okay, there's not much in there. And if I look right here, okay, this is my cell membrane. So my cell wall structure stays the same. But when I compare it to, like, for example, my animal cell is starting to shrivel. The same thing happens down here. Okay, the cell membrane pulls away from the wall. Okay, and if we're talking about the plant's going to start to wilt. And so this process of that cell membrane pulling away, that's called plasmolysis. Okay, so remember, lysis means to break down. Okay, plasmo, we're talking about that cytoplasm. Okay, so the cytoplasm is breaking down and the cell membrane is shrinking. Remember, cytoplasm kind of helps keep everything in place. So lastly, in a hypotonic solution. Okay, surprisingly, this is actually what plants like. Okay, plants like a hypotonic solution. Okay, so again, higher concentration of solutes on the inside than the outside. Okay, so water flows in. And in this case, the cell is turgid. Okay, so what that means is that my central vacuole, okay, it's all pressed up here against this membrane and against the wall. And so it's all full of water. This is what plants actually like. And that cell wall is going to prevent it from exploding. So these are different scenarios for different solutions for plant and animal cells. Okay, so that's why, for example, in isotonic solution, when we're in equilibrium, we're not in pure equilibrium with water. Okay, so for those of you that wear contacts, I actually found this in my bag, so it's kind of funny. So if I read, it says like it contains sodium citrate, sodium chloride, boric acid, things like that. Okay, and this is going in my eye. Okay, this is going in my eye with my contacts. But the reason why it has those solutes in it is because, you know, we're not in equilibrium with water. We do have some salts. And it's the same thing like if you have to go to the hospital and get an IV bag, you need some salts. All right, so last thing we're going to talk about, this one should be short because we just talked about osmosis, is facilitated diffusion. So facilitated diffusion is the diffusion of molecules or ions through a protein channel or a transport protein. So same thing, our goal is to get to equilibrium, okay? But it's just, you know, these molecules are too big, they're not going to flow through. Usually the molecules that do this are going to be things like sugar molecules, amino acids, ions, and then, you know, molecules that are small to medium size. Okay, so those are usually what moves through. And so when this happens, your ion channel is open or the transport protein is active. That way molecules can move in both directions. Remember, we're still talking about passive transport. So, for example, when I'm looking at this diagram, I have more chlorine ions at the top of my membrane, so they're going to be moving down. I have more potassium ions at the bottom, they're going to be moving up. Okay, so my example, chlorine moving down, potassium moving up. Okay, so very important, each channel is molecule specific. Okay, so I can't have potassium ions moving through this chlorine channel. Okay, vice versa over here. So what changes the rate of facilitated diffusion? Well, the same thing as the last stuff. Okay, so temperature concentration, and the number of proteins embedded in the membrane. Okay, the more proteins you have, the more stuff will move in and out. So this was for our channels. I said up here that we could have transport proteins. What are those? So I'm going to come down here and look. Okay, so we have some that are gated. Again, I realize I forgot to color my diffusion and my molecules here. So we have some channels that are gated. That open and close based on concentration. Okay, so an example of this, okay, so in our nervous system, okay, we have membrane potential. And we're going to talk about this when we talk about our nervous system. Potential, does it have an A? It does. Okay. So essentially we have this electric charge. It's going to build up outside, and then what ends up happening is this charge is going to bind, and depending on whether that charge is present or absent, those molecules will flow through. Okay, so a change in charge allows the flow of ions. So this would be an example of a transport protein. You have to have that charge there in order for this gate to be closed. If there's no charge there, then that gate's going to be open, and so now I'm going to have sodium ions travel through. So same thing affects this, temperature, concentration of ions, but the only thing is that it's monitored for how many ions will travel through. So hopefully this helps. We're going to be practicing this on Monday, so make sure you're ready.